Okay, welcome back. This is the January 20th Part 2 lecture. We left off last time talking about hormones and where the receptors are located for those hormones. We mentioned a couple of places. Those receptors might be in the nucleus of the cell, right on a DNA strand, for instance, or those receptors might be embedded in the outer cell membrane, sticking outward to bind to some hormone on the exterior of the cell. And then also hormones receptors might also be in the cytoplasm of a cell. And typically in those cases, this is for lipid-soluble hormones. They're able to get through the cell membrane and into the cytoplasm. And so when those lipid-soluble hormones bind to those receptors in the cytoplasm, the hormone receptor complex then migrates into the nucleus, ultimately binding to the DNA. Okay, so that's where some of these receptors are located. Now, what happens when hormones bind to receptors? What are the mechanisms by which these hormones work? And there's two big categories of hormonal action that I'm going to talk about. And the first category is that hormones can work by direct gene activation. So I'm going to give that one a number. Hormones work by, I'll say, number one, direct gene activation. Okay? If you want to see this in action, check out slide number 16 in your PowerPoint. It's an enlarged view of a cell in a target tissue, so it's a target cell. And as you can see, there's a nucleus in there in purple. The cytoplasm is in a yellowish color, and the extracellular fluid, the interstitial space, is in blue there. So if you look at the top diagram there, you see a hormone. Maybe it's a steroid hormone, but some lipid-soluble hormone dumped out of the bloodstream, streaming through the interstitial space, bumps into the outside of the cell and slithers right through that phospholipid bilayer because it's lipid soluble. It can easily move through the phospholipid bilayer, and so it makes its way into the cell. And as you can see on slide 16, what happens next is the receptor for that hormone might be sitting there in the cytoplasm, and the hormone will bind to that receptor, forming what's called a receptor hormone complex. And then that receptor hormone complex there in the cytoplasm moves into the nucleus of the cell, and you can see it then binds to a particular location, specific binding to some specific location on the DNA to typically either turn on or turn off the transcription of a gene. And ultimately, influencing transcription of genes means we influence which messenger RNA strands get made and ultimately which proteins get made in that cell. And that's a really strong way to control activity, is by dictating what proteins should be made in a target cell. For instance, enzymes are proteins, so a hormone could potentially influence which enzymes get produced in a cell. So this whole thing is called direct gene activation, and I'll just write it out here, but this is slide 16. Hormone binds to a receptor. Again, either in the cytoplasm or in the nucleus. And regardless of which of those two it is, nucleus, regardless of which two is the case, that hormone receptor complex then binds to a portion of the DNA. So that hormone receptor complex then binds to a specific part of the DNA. And as I mentioned a minute ago, influencing which genes get transcribed and translated. Influencing which genes get transcribed and translated. Okay? Maybe you've taken a genetics course and you know that right next to genes for certain gene products is often a promoter region. And in those promoter regions, if certain chemicals bind to those promoter regions, that triggers the onset of transcription, the making of a messenger RNA from a DNA strand in a particular gene locus. So this is called direct gene activation because these hormones are directly activating particular genes to be expressed in a target tissue cell. And while slide 16 shows the hormone receptor located out in the cytoplasm, I do want you to know that in some cases the receptor is already waiting in the nucleus. So for instance, for thyroid hormone, the thyroid hormone receptors, those that work by direct gene activation, are already in the nucleus, always bound to the DNA, just waiting for thyroid hormone to stream on in and bind. I should also mention that in some cases, the cytoplasmic receptor sitting out there, in the absence of hormones, is often bound to still other molecules, chaperone-like molecules, molecular chaperonins. These are proteins bound to the receptor. And the binding of the hormone to the receptor bumps off the molecular chaperonins and allows the hormone receptor complex to then move into the nucleus. So if you want an example of this for a particular hormone, consider the hormone testosterone. Testosterone means it's a steroid, means it's lipid soluble. Well, we know that testosterone hormone works by direct gene activation, and one of the things testosterone does is builds up muscle bulk, makes skeletal muscles larger. So you can imagine testosterone as a hormone released from the testes into the bloodstream, makes its way to skeletal muscle, jumps out of the bloodstream, slithers into skeletal muscle cells, binds to some receptor, ultimately receptor hormone binding to the DNA next to maybe the genes for actin and myosin, those two myofilaments that make up most of the contractile machinery of skeletal muscles. So this would be, for instance, how testosterone can create long-term changes in literally the amount of actin and myosin and the number of sarcomeres in and the size of the sarcomeres in those cells. So muscles bulk up literally as a result of direct gene activation from testosterone and similar hormones. So one mechanism that hormones use to influence target tissues is by direct gene activation. But what about for hormones that are not lipid soluble, that can't easily pass through the cell membranes of target tissue cells? They bump into the outside of the cell, and that's as far as they can get. Well, those hormones, the water-soluble hormones, don't work by direct gene activation, but rather they work by second messenger systems, or second messenger mechanisms. So I want to describe these second messenger mechanisms and give you a couple of examples of them. And I would say let's start by looking at slide number 17 in your PowerPoint presentation. It shows a nice overview of what happens when hormones are working via second messenger systems. Okay, so again, this is used by hormones that are water soluble, used by hydrophilic hormones, water soluble hormones. They are not lipid soluble, so they can't get into the cell. The best they can do is bind to some receptor on the surface of the cell. So let's draw a little picture of this if we can, to think about how this might work, and you can also look at slide number 17. So here's a little capillary here, like so. Blood coming in this direction, and you have maybe some hormone. Maybe it's shaped like a triangle. Maybe it's adrenaline hormone, epinephrine. That's a water-soluble hormone. And then you have a target cell right next door here, like so. That's your target tissue cell with its cell membrane outlined there. So what happens? This hormone that I'm drawing as a green triangle, again, one example might be epinephrine, but there are several other examples of hormones that work in this way. But here comes epinephrine, goes through the wall of the capillary, approaches the receptor on the surface of this target tissue and binds to that receptor. And it binds because its shape nicely matches the shape of the receptor. And so when it binds, it stays there for a little while. And in this particular second messenger example, this is the cyclic AMP mechanism. I'll just write that here. Cyclic AMP mechanism of second messengers. So what happens here? Well, you got hormone binds to receptor on a cell surface. And that initiates a cascade of events. Okay? First of all, there's a G protein, regulatory G protein, right next to the receptor. So when the hormone binds to the receptor, that activates a neighboring regulatory G protein. It's a regulatory G protein, it's called. And once that G protein, which is this guy right here, I'll label it here. Once that G protein is regulated, or activated, I should say, it releases itself from the receptor and migrates next door to where an enzyme is located. And the enzyme that I'm talking about is adenylyl cyclase, and I'm going to draw it like this in blue. Okay? So my little symbols here, the little black dot is the G protein, and my little blue arc-shaped structure here is a membrane-bound enzyme called adenylyl or adenylate cyclase. Adenylate or adenylyl, you can say it either way, cyclase enzyme. Okay, so what happens? Hormone binds to receptor, activates a G protein, which gets released and moves over to adenylate cyclase. In the membrane there. Okay? So that adenylate cyclase enzyme then gets activated. So the binding of the G protein to it activates that adenylate cyclase enzyme. And then, by the way, in slide number 17, the adenylate cyclase is shown in sort of a pinkish purple color in that drawing. Okay? But anyway, the G protein gets released, moves to adenylate cyclase, and activates it. Okay, so then the question is, well, what does adenylate cyclase do? Well, adenylate cyclase enzyme actually converts ATP, our old friend ATP, into something called cyclic AMP. And that cyclic AMP is the second messenger that will have subsequent effects in this cell. So adenylate cyclase, once activated, then converts ATP adenosine triphosphate, into cyclic AMP, cyclic adenosine monophosphate. And it's that cyclic AMP which is the second messenger in all of this. Now, before we move on to the next step in this, let me just backpedal a little bit and just talk about this G protein. So this G protein actually has GTPase activity in it. In other words, it's a protein that also acts as an enzyme. And what happens is that G protein can bind to GTP, guanosine triphosphate. And when it binds to, when guanosine triphosphate or GTP binds to it, it displaces a guanosine diphosphate on there. That's what actually releases this G protein from its connection to this first receptor. And as I said, it sort of floats through the phospholipid bilayer over to and bumps up next to adenylate cyclase. Now, when it bumps up next to adenylate cyclase, the GTP that's bound to the G protein gets cleaved into GDP and a phosphate, and the energy released from that is actually what activates the adenylate cyclase. So there's a little bit more to this G protein. I just wanted to give you that little extra detail there. So now, adenylate cyclase converts ATP into cyclic AMP, which is a second messenger. We call it a second messenger. Who's the first messenger? Why do we call this one a second messenger? Well, the first messenger was the hormone. And the hormone binding to this receptor on the outside of the cell is sort of like someone going up to someone's door and ringing the doorbell. And the ringing of the doorbell then initiates a whole flurry of activity inside the house. In this case, a flurry of activity as a result of the formation of cyclic AMP as a second messenger. So how then does this cyclic AMP act as a second messenger? Well, this is outlined on that slide 17 again. And what cyclic AMP does is then activates enzymes that are located in this cell called protein kinase enzymes. So cyclic AMP binds to and activates various protein kinase enzymes already waiting in the wings here in the cell. Protein kinase enzymes. Maybe you've learned in other courses what a protein kinase enzyme does. A protein kinase enzyme specializes in phosphorylating stuff. By that I mean it attaches phosphate groups to other molecules, which in turn activates or deactivates those other molecules. So cyclic AMP binds to and activates protein kinase enzymes, which those enzymes in turn phosphorylate other molecules in the cell. Phosphorylates other molecules in the cell. And what other molecules are we talking about? Well, it depends on the target tissue. And one target tissue might have a certain set of protein kinase enzymes ready to go that can phosphorylate some other type of protein. Another target tissue might have protein kinase enzymes that can phosphorylate still other proteins. So in other words, the generic way that this happens is via cyclic AMP, but which particular protein kinases and which particular phosphorylated molecules develop depends on the target tissue. So to fill out this diagram here, the cyclic AMP here, as a second messenger, activates inactive protein kinases. They get transformed into active protein kinases. What do? Those are enzymes that phosphorylate other molecules to create further effects in the cell. So, phosphorylating these other molecules triggers further effects in the cell. Okay? So if we round out this picture then, we see that inactive protein kinases getting converted to active protein kinases creates further phosphorylations and further effects in the cell. So one question you might be asking, I asked this when I first learned about this, is why so complicated a process? It's the picture I just drew a minute ago. There are a lot of steps in this. Hormone binds to receptor, receptor activates G protein, binds to GTP, GTP gets converted to GDP, and that G protein conversion helps activate another enzyme, which then converts ATP to cyclic AMP, then activating protein kinases, which phosphorylate other things. Why so complicated? Why does this have to be such a complicated process, multi-stepped? Well, I can think of two distinct advantages to having this second messenger system be a multi-stepped process. So what are the advantages of this being a multi-stepped process? Why not just have one step? I can think of two things. Number one, it allows for more places where we might tweak this mechanism and fine-tune it, control it. So this allows for control of the process, better control of the process. I'll give you an example in just a minute of how that might play out. But this allows for better control of the process. There's lots of places where we can tweak this, okay? And secondly, because many of these steps actually involve one thing activating an enzyme, which triggers another chemical reaction, which activates more enzymes. Remember how fast enzymes work? A single enzyme can convert a reaction 1,000 times over in a second. So if you think about it, if you had just one hormone binding to a receptor, activating just one G protein, activating just a single adenylate cyclase enzyme, well, then that might convert 1,000 ATPs into cyclic AMPs in 1 second. Now you've got 1,000 cyclic AMPs, and they're binding to 1,000 protein kinases and activating them. And then the protein kinases start phosphorylating things. So with each step, you're exponentially increasing the number of activated molecules. 1,000 times 1,000, that's 1,000,000. And so one single molecule ringing the doorbell on the outside of that cell, binding to one receptor, can have very potent effects inside when it works by a second messenger mechanism. So again, multi-stepped process, the sequential activation of one enzyme after another the sequential activation of one enzyme after another in this process allows for exponentially more potent effects of that hormone. So you don't need a lot of the original hormone, okay? You don't need a lot of that hormone because of the way this works. All I got to do is have one person go to that door and ring the doorbell, and maybe 10 or 20 other people in the house start scurrying about as a result of ringing that doorbell. One single hormonal molecule can bind. For instance, glucagon. We'll learn about the hormone glucagon. One of the things that glucagon hormone does is raise your blood sugar level. And it's been estimated that one glucagon hormone molecule has, through a second messenger system, the ability to add 100 million glucose molecules to your bloodstream. That's incredible. And so a multi-step process in which each step involves one enzyme activating another, allows for a very comprehensive, full-on, potent response with the help of really just a small number of original hormone molecules. Now, let me give you an example of the first advantage I mentioned here, better control of the process. One thing that's not shown on slide 17 is that cyclic AMP, that second messenger, can get deactivated. So you have active cyclic AMP, it can actually get deactivated into what's called 5' AMP. And the enzyme that does that is an enzyme called phosphodiesterase. And there's actually several sub-varieties of phosphodiesterase, several types, type 1, type 2, type 3, etc. But the enzyme's called phosphodiesterase. And that enzyme, I'll put a plus sign here, meaning it speeds up this reaction to convert active cyclic AMP into inactive 5' AMP. So it's a way to tweak the system. We can slow down the activity because we have these steps that are then vulnerable to being turned on or turned off by these additional enzymes. So if we want to add that to our original picture that I showed you earlier, let me just do this. You could add here that cyclic AMP can get converted to the inactive 5' AMP. And the enzyme that does that, I'll label it PE for short here. PE is shorthand for phosphodiesterase. And it turns out that that's a very important clinical point to make. Why is that so important clinically? Well, the clinical relevance of this, knowing about phosphodiesterase, the reason that's clinically relevant is that there are a group of drugs called phosphodiesterase inhibitors that inhibit the action of that enzyme. These are phosphodiesterase inhibitors. All right? And these are drugs which block the action of phosphodiesterase. Why might we want to do that? Why might we want to block the action of phosphodiesterase? Well, what would happen if you block the action of this inactivating enzyme? If we block the inactivation of cyclic AMP, the net effect of that is to keep cyclic AMP levels high in that target cell. All right, so by blocking this enzyme, they block the action of phosphodiesterase, thereby keeping cyclic AMP levels high in the target tissue, keeping the concentration of cyclic AMP high in that target cell or target tissue. So, in other words, enhancing the effect of whichever hormone it was that originally bound and set this whole process into motion. So enhancing the potency and the effect of the original hormone triggering this process, whatever that hormone might have been. Its action gets enhanced. All right? So one example of a hormone that might trigger the cyclic AMP mechanism is epinephrine. So the original hormone, epinephrine, its effects would get enhanced if you gave someone a phosphodiesterase inhibitor. What are these drugs? What are examples of these drugs? Theophylline is an oral drug given by mouth. It's a phosphodiesterase inhibitor. Aminophylline, closely related chemical, is also a phosphodiesterase inhibitor. It should be a Y, not an I. Aminophylline. And caffeine. is actually an example of a phosphodiesterase inhibitor. All three of these drugs are examples of methylxanthines. That's the organic chemistry term for them. Methylxanthines, which block the effects of phosphodiesterase. And so they keep cyclic AMP levels high in these cells. So if you think about it, a hormone that uses the cyclic AMP second messenger system, its effects will be enhanced by these mechanisms. One such hormone is epinephrine. So these phosphodiesterase inhibitors, they work to enhance epinephrine's effects. They enhance the effects of epinephrine, or adrenaline as it's commonly called. Epinephrine and adrenaline mean the same thing. It's the same chemical. We sometimes call it adrenaline. But since adrenaline or epinephrine works by cyclic AMP messengers, second messengers, their effects are going to be enhanced. So think about it. What did we learn about last semester regarding epinephrine? It's a hormone of the fight or flight response. It triggers a lot of the fight or flight response activities during times of emergency. So what does that mean? Well, for instance, it will cause the smooth muscle around your small airways, the so-called bronchioles in your lungs, to dilate, to relax and dilate. So one of the normal effects of epinephrine is to cause bronchiolar dilation. That's the real term for it, bronchiolar, even though most people call it bronchodilation. It really should be called bronchiolar dilation of the small airways in the lungs. So if those small little airways dilate, you can get more air in. So what sort of person might be given a drug called theophylline or aminophylline? Well, how about an asthmatic? So this is useful in the treatment of asthma, to bronchodilate. So a life-threatening condition, asthma, in which your small airways are too constricted down, can be treated somewhat effectively with either oral theophylline or, in a severe case in the emergency room, IV aminophylline, intravenous aminophylline, as a result of this. And so that's just one example of how phosphodiesterase inhibition might be useful clinically. Now, I mentioned caffeine. If you are a caffeine drinker, you're probably well aware that if you drink too much coffee and take in too much caffeine, what kind of symptoms do you get? Heart racing, sweating, irritability, feeling on edge, maybe even palpitations. All of this because one of the reasons for this is because caffeine is acting as a phosphodiesterase inhibitor in various target tissues, keeping levels of cyclic AMP high in those tissues, enhancing the effects of those epinephrine hormone molecules coming from the bloodstream into those tissues. Another thing I should mention here is that this whole conversation is about cyclic AMP as a second messenger. I just want to let you know that cyclic GMP, cyclic guanosine monophosphate, can also act as a second messenger in certain tissues, and cyclic GMP works in a parallel way to cyclic AMP. And also, cyclic GMP can get inactivated to 5' GMP by guess who? Phosphodiesterase enzymes. And if we want to enhance cyclic GMP levels in cells, we could inhibit those phosphodiesterases. In fact, in the last 20 years, drugs that have come onto the market to treat erectile dysfunction, drugs like Viagra, which is sildenafil, and Cialis, which is tadalafil, these are actually drugs that work by blocking phosphodiesterase type 5 in erectile tissue. And as a result of blocking that enzyme in erectile tissue, it keeps cyclic GMP levels high in those tissues, which relaxes blood vessels and allows more blood to flow into those erectile tissues. So it's a way of treating erectile dysfunction, and it utilizes our knowledge of second messenger systems, in that case, the cyclic GMP second messenger system. Now, I'd like you to take a look at the next slide in your PowerPoint. It's slide 18. This one isn't in your textbook, it's in an older edition, but I like this slide because it shows what we've just talked about, the cyclic AMP second messenger system, how it works, but it also shows on the left side all the different hormones that use cyclic AMP as a second messenger. So at the top of that list, catecholamines, what are we talking about there? Epinephrine and norepinephrine. But also other hormones we're going to learn about in this unit, like ACTH, FSH, LH, glucagon, PTH, etc. They all use cyclic AMP as a second messenger. So you might ask, if all of these hormones use the same second messenger, wouldn't they all have the same effects in the body? And of course the answer is no. Catecholamines have different effects from ACTH or FSH or any of the others because those hormones bind to different receptors on the surface of the target tissues. So epinephrine might bind really well to receptors on the surface of the smooth muscle cells around your airways, but might not bind so well to other target cells that are targets of a different hormone. ACTH binds to one set of receptors, catecholamines bind to another, FSH to another. So it's where the receptors are and which receptors are bound to that dictates where the target tissue effects are going to be. So in summary, all these different hormones, they can all use the same cyclic AMP second messenger mechanism. It doesn't mean they have the same effects, because it's the availability of a receptor on the cell surface that dictates whether the hormone's going to have any effect at all in that target cell. Now, I'd like you to turn next to slide number 19 in your textbook, and we're going to finish up today with the discussion, or introduction at least, of another second messenger system, and I'm going to call it the calcium calmodulin second messenger. Calcium calmodulin second messenger. And slide 19 shows how this works. Now, the general concept is similar to the cyclic AMP and cyclic GMP mechanism. If you look at slide 19, we're looking close up at a target tissue cell membrane with a phospholipid bilayer there. You see a hormone in the upper left binding to a receptor. Once again, activates a G protein. The G protein activated, released, swims next door through the phospholipid bilayer, where it then activates an enzyme. And that enzyme is called phospholipase C. And when that enzyme gets activated, that phospholipase C enzyme speeds up a chemical reaction. It takes PIP2, splits it into DAG and IP3. Now, PIP2 is phosphatidylinositol biphosphate. DAG is diacylglycerol, and IP3 is inositol triphosphate. So basically, this enzyme phospholipase C gets activated. It splits a phospholipid molecule called PIP2 into DAG and IP3. Now the DAG, the diacylglycerol, remains in the cell membrane there, but activates a neighboring inactive protein kinase C. So here we go, activating another protein kinase called protein kinase C. That active protein kinase C then triggers phosphorylation of other substances in the cell and various effects. But what about the other split product? In addition to DAG, you also have inositol triphosphate, IP3, formed. And look what happens to that. That gets released into the cytoplasm, moves to the endoplasmic reticulum, where it then triggers the release of calcium ions from the endoplasmic reticulum out into the cytoplasm. And the calcium ions spilling out into the cytoplasm bind to a protein that's already out there called calmodulin. And that calcium calmodulin complex then triggers a bunch of responses in the cell. So we see once again a multi-stepped cascading mechanism where just one or small number of hormones ringing the doorbell on a receptor outside the cell, creating a flurry of activity on the inside of the cell and allowing for an amplification of the effects for a very comprehensive response. There's a list of hormones in the lower left of that slide, just to give you an idea of some hormones that use this calcium calmodulin second messenger. And look, lo and behold, catecholamines. Epinephrine in certain cells triggers this second messenger system. So it's possible for the same hormone to trigger one type of second messenger cascade in one cell and another type of second messenger cascade in another cell. So this is just another example, and I'm going to put in the notes here, refer to slide 19 because I'm not going to rewrite all that. But be sure to take a look at and know slide 19 and let me know if you have any questions about that. And with that, I will stop here and we'll see you guys at the next lecture. Take care.